Hello and welcome to Zoology 142. This is our first lecture on the reproductive system. Today we're going to start talking about meiosis and also talking about the male reproductive system. Mainly because of the two reproductive systems, the male reproductive system is the simplest. So after you complete today's lecture, you should understand the difference between mitosis and meiosis. You should be able to identify key parts of the male reproductive anatomy and also to discuss the role of testicular cells in hormone production and also spermatogenesis. You should be able to describe the hormonal axis necessary for production of testosterone and also describe stages of the male sexual response. And finally, you should be able to recognize and describe key disorders of the male reproductive system. So because this is a college class, I'm not going to talk about the birds and the bees. I assume that all of you know how sexual intercourse works. We're going to focus first on sexual reproduction. And sexual reproduction involves the production of offspring by the fusion of gametes, that is, eggs or sperm. Gametes are haploid sex cells which are produced in the gonads, that is the ovaries or the testes. And by haploid I mean they have half the number of chromosomes that a normal body cell would have. For example, a normal body cell would have 23 pairs of chromosomes for a total of 46, whereas either a sperm or an egg is going to have 23 chromosomes. So it has half the number of chromosomes of a normal cell, so we say that it's haploid. So in my face-to-face lectures, I like to start the discussion of sexual reproduction with a question, and that is, why exactly do we have sex? Well, I've gotten several answers over the years, and I'm going to share a few of them with you now. One of the more common answers I get is to perpetuate our species. That is, we all recognize that we have a finite lifespan, and the only way to perpetuate the human species is to reproduce and have offspring. But there's a little bit of a flaw in that answer. Specifically, not all organisms reproduce sexually. For example, sea anemones, which you see here, can just divide and produce two different organisms without sexual reproduction. The same happens in plants. Banana plants can produce keikis without any kind of sex. Another common answer I get is to pass on our genes. And yeah, this is a very noble cause that we want to pass on our genes to future generations. But is that really the reason we have sex? Now remember that humans aren't the only mammals that have sexual reproduction. So next time you see a couple of dogs going at it on the street, do you really think they're sitting there going, "Must pass on my genes"? No, there's something else that's going on, some reason other than passing on genes, that they're having sexual intercourse. Okay, another common reason is that we want to have babies, and the only way to have babies is to have sex. And you can't really argue with that, because there is some truth in it. You cannot have a baby without having sex. But a lot of people have sex without wanting to have a baby. Some of the more scientifically minded of you in the class might say, well, sexual reproduction allows for greater genetic variation. And that's true. Among organisms that reproduce asexually, we tend to think that they're at greater risk of extinction because they're not able to adapt to changing environments. And by having sex and exchanging genetic materials, we're enabling ourselves to recombine our genes with those of others, which should allow us to more easily adapt to changing environmental circumstances. So that's certainly a strong argument for having sex. And of course, some people will say, well, we have sex because sex feels really good. And this is true. Sex feels good for humans, it feels good for dogs, it probably feels good for all sorts of mammals, and even maybe some invertebrates. So why does sex feel good? Well, sex feels good because it helps us to do all these other things earlier in the list. Because sex feels good, we tend to have sex. And because we have sex, we tend to perpetuate our species, pass on our genes, and also allow for greater genetic variation. So all of these answers are correct. The one at the bottom, because it feels good, is the proximate reason that humans have sex. That is the reason we do it without really knowing what we're doing. Whereas the ultimate reasons are the ones above, perpetuating the species, passing on genes, and allowing for greater genetic variation. So now that we started the lecture with a decidedly sexy topic, we're going to switch streams and talk mainly about a very non-sexy topic, that is cell division. Before we can understand how cellular division works, we need to talk about the human chromosomes. A normal body cell has 2 copies of each chromosome and are therefore said to be diploid. This means that we have 1 copy from our mom and 1 copy from our dad. And remember, these chromosomes contain genes which are coded in our DNA, and each gene is a different recipe for a different type of protein. And obviously you're getting slightly different versions of these genes from both your mother and your father. And again, this allows for greater genetic variation, which helps our species to adapt to changing circumstances. Now we call these two chromosomes that are inherited from both our mother and father a homologous pair. Now we have 23 homologous pairs of chromosomes in our body, and each member of the pair carries slightly different information than the other member. We also have the sex chromosomes, or the X and Y chromosomes. If you're female, you have two X's, and if you're male, you have an X and a Y. In contrast to normal body cells, gametes, that is, eggs and sperm, have only one copy of each chromosome and therefore are said to be haploid. The way that we create haploid cells is through a process called meiosis. So meiosis is a type of cellular division. You're probably already familiar with another type of cell division called mitosis. Mitosis is a type of cell division which results in the production of two genetically identical diploid daughter cells. So if you take a look at the figure at the right, you see we have one mother cell, and that cell will divide to make two daughter cells. And these daughter cells have two copies of every chromosome, so they're diploid, and the cumulative genetic information in these two cells is exactly the same. And so mitotic cell division is probably the most common type of cell division in the body, and two examples are growth, that is, as you grow larger, the more cells you add is through the process of mitosis, and also wound repair. If you cut yourself and that wound heals over the period of days and weeks, that wound repair is through the process of mitosis, which gives us cells that are genetically identical to one another. If you would like to refresh your memory about mitosis, please click at the link at the bottom of the screen. Now the second type of cell division, which happens only in the ovaries and testes, is meiosis. And this is the type of cell division that leads to the formation of genetically distinct haploid daughter cells. That is, each of these cells carry half the number of the chromosomes of the parent cell, and each cell, when compared to each other, are genetically different. We're not going to go through the mechanics of meiosis in this lecture, so you should review this in your textbook, and you may also want to click the link at the right to see an animation of meiosis. So remember that our gametes, the sperm and the egg, are both haploid. That is, they contain half the number of chromosomes of a normal body cell. Now during fertilization, we get these two haploid cells together and they form a zygote. So a zygote is a diploid cell caused by the fusion of the sperm, which has 23 chromosomes, and the egg, which also has 23 chromosomes. So the zygote will have a total of 46 chromosomes. Another name for a zygote would be a fertilized egg. Once the zygote is formed, it will then divide mitotically to form a fetus or eventually a baby, and this will involve over 10 trillion cellular divisions. But because we're involving mitosis here, it's important to realize that all the body cells in this baby, save the ovaries or testes, are going to be genetically identical to one another. Now here's another question. What do you think would happen if the gametes did not go through meiosis, that is, they did not become haploid? Well, let's take a look at the answer. Imagine that we had a diploid sperm with 46 chromosomes and a diploid egg with 46 chromosomes, just like a normal body cell. When we fuse these two gametes together to make a zygote, we're going to end up with something that's tetraploid. That is, it has 4 copies of every chromosome. And remember, a normal body cell only has 2 copies of every chromosome. Now, you probably already know that some genetic disorders, for example, Down syndrome, result because we simply have 1 extra chromosome, and this can cause profound and life-changing defects in the human body. So imagine what it would be like if we had 2 extra of every chromosome. It basically wouldn't be possible to bring a fetus to term if it was tetraploid. So that's the reason why we have to go through the process of meiosis in order to make our sperm and our egg. The sperm and egg must both be haploid so that when we make the zygote, it will be diploid. Okay, so now that we've had an overview of the process of meiosis, we're going to start talking about the male reproductive system. Now, I'm not slighting anybody here by starting with males. We're simply starting with the male reproductive system because it's the simplest of the 2 reproductive systems. So the functions of the male reproductive system include the production of gametes, that is sperm, the production of seminal fluid, and also the delivery of that seminal fluid and sperm into the female reproductive tract. And finally, another function of the male reproductive system is to produce hormones, for example, testosterone. So as I said before, the male reproductive tract in comparison to female is relatively simple. It consists of the testes, which are our gonads, the scrotum, which surrounds the testes, the various ducts, which help to transmit the sperm and seminal fluid into the penis, the duct system, which includes the vas deferens, which helps to transmit the semen and sperm into the urethra. It also includes the accessory sexual glands, for example, the prostate gland, and also the penis, which is the organ of intromission. And we'll talk about that in just a minute. So we're going to start out our discussion of the male reproductive anatomy with the scrotum. The scrotum is an outpouching of skin from the abdominal wall, and it contains muscle tissue that allows the testes to thermoregulate. The reason is because sperm production cannot go on at normal body temperature, that is 98.6. It has to be about 3 degrees lower than surrounding body temperature. And that's the reason why the testes are hanging outside the body in this rather precarious position in this small sac of skin. And this sac contains 2 different muscles that help it to thermoregulate. The first of these is the dartus muscle. The dartus muscle is the muscle in the skin of the scrotum, and this helps to crinkle up the scrotum when it's very cold outside and helps to hold the testes close to the body. The cremaster muscle is a larger muscle that attaches to the testes and unites to the pubic ligament. And again, it helps to draw the testes up closer to the body when it's really, really cold. In fact, rabbits can use the cremaster and other muscles to actually pull the testes inside the body when they're fighting, which seems like a really great adaptation. Now, if you ever saw the Seinfeld episode about shrinkage, you know that the scrotum and penis will reflexively contract if it's cold outside. And that's what happened to poor George Costanza. He came in from, I don't know, a cold swimming pool or something like that, and somebody caught him undressing and, was laughing at him because things were a lot smaller than they should be. And that's because the dartus muscle and the cremaster muscle are helping to conserve the heat in the testes and hold them up close to the body. If you'd seen the same person, let's say if they walked out of a sauna, the scrotum would be much more relaxed and the testes would be hanging well away from the body in order to properly thermoregulate. So the testes are the male gonads, and the gonads are the things that actually produce the gametes, in this case the sperm. And not only do we produce sperm, but we also produce the hormone testosterone, which is responsible for spermatogenesis and also for male secondary sexual characteristics. Now, it's important to realize that the testes actually develop inside the body near the kidney, and they descend through the inguinal canal during the last months of fetal development. You can see here that attached to the testis are the vas deferens, which helps to transmit sperm into the urethra, the epididymis, which is a storage place for sperm, and also the testicular blood vessels and nerves. We have a large papilliform plexus which transmits oxygen-rich blood to the testis and also drains the testis via venous return. It's interesting that these blood vessels, the arteries and veins, run very close to one another, and this allows for countercurrent exchange. Basically, the warm arterial blood that's coming down from the body cavity is cooled off by the return of venous blood, and again, this helps to keep the temperature of the testes about 3 degrees lower than the normal body temperature in order to promote ideal spermatogenesis. Now, the testes are surrounded by 2 tunics or layers. The first of these is the tunica vaginalis, and this is the most superficial tunic of the testis. Below this is something called the tunica albuginea, and the tunica albuginea is made up of fibrous connective tissue, and it basically invades the inside of the testis to divide it into lobules. There's approximately 25 lobules in each testis, and each one of these lobules contains 1 to 3 seminiferous tubules. These are the functional units of the testis, that is, these are the locations where sperm are produced, and the process of producing sperm is called spermatogenesis. So here's a histological section of the testis and the seminiferous tubules. In the middle of the screen you can see one seminiferous tubule, and on the outside of that tubule you can see something labeled letter G. Letter G indicates a spermatogonia. These are the most immature cells that will undergo mitosis and later on meiosis to form the more advanced stages of spermatogenesis. If you look at Z, you can see a spermatozoa. The spermatozoa are the most advanced or developed gametes within the male reproductive tract. You can see that they have a well-developed tail and that they line the periphery of the lumen of the seminiferous tubule. Now, once they are mature, they will basically break away from the side of that wall and be swept through peristaltic contractions up to the epididymis and eventually through the vas deferens. On the outside of the tubule, you can see something labeled letter I. Letter I indicates something called an interstitial cell, and another name for this is a Leydig cell. And we'll talk about the function of the Leydig cell in just a minute. So here's a drawing of a single part of the wall of a seminiferous tubule, so you can see the different types of cells that are present. We have the spermatogonia, which are diploid, primary spermatocytes, which are also diploid, and finally secondary spermatocytes, spermatids, and also spermatozoa. Each of these is haploid, that is, they've undergone the process of meiosis. And spermiation is actually the process where the spermatozoa are released from the Sertoli cells and enter the lumen of the testis, which would be at the bottom of the screen, and again are eventually propelled through the seminiferous tubules to the epididymis, where they will be stored and later ejaculated. Now, in addition to the spermatogenic cells that are in the seminiferous tubules, we have two other cells that are very important for spermatogenesis. The first of these is something called the interstitial cell or Leydig cell, and the Leydig cell is actually located outside the seminiferous tubule, and that's why we call it an interstitial cell. The function of the Leydig cell is to produce testosterone, and testosterone is necessary for spermatogenesis. It's also necessary for the production of male secondary sexual characteristics, which we'll talk about in a few slides. The other cell that you see here is something called a Sertoli cell. A Sertoli cell is also called the sustentacular cell, or the mother cell, and its job is to nurture the maturing spermatozoa and spermatogonia as they go through the process of meiosis. Now, the way that the Sertoli cells nurture the spermatogonia and spermatozoa is by production of nutrients and essential endocrine signals that help to set the timing for sperm production. They also help to phagocytize faulty cells and also digest cell fragments that are produced in the process of meiosis. And finally, they produce inhibin. Inhibin is an important hormone that helps to basically stop the production of testosterone once there's enough spermatozoa within the seminiferous tubules. So this is just an overview of the process of spermatogenesis. Spermatogenesis involves both mitosis and meiosis. We're going to start with a diploid cell called a spermatogonium. The spermatogonium is going to grow into something called a primary spermatocyte, which is also diploid, and this is going to undergo meiosis 1. Meiosis 1 is going to produce secondary spermatocytes, which are haploid. We're then going to go through a second set of divisions through meiosis 2, and this is going to produce haploid spermatids. And these haploid spermatids are eventually going to get tails and mature into good old haploid spermatozoa. Now, the thing you should notice here is that for each spermatogonium, we get 4 spermatozoa, so it's quite a bargain. But it's important to realize that each of these spermatozoa are haploid, and they're also genetically distinct from one another. Now, remember that the least mature cells are going to be found at the periphery or on the outside of the seminiferous tubules. So here we have a little bit of a section of a seminiferous tubule. The outside is toward the bottom of the screen, and the inside or lumen is towards the top of the screen. At the very bottom, you can see our diploid spermatogonium. These are the most immature cells within the testis. The diploid spermatogonium are going to undergo a mitotic division to produce a primary spermatocyte. Just like the spermatogonium, the spermatocyte is diploid. That is, it has 2 copies of each chromosome, or has a total of 46 chromosomes. The primary spermatocytes will undergo meiosis 1 to form the haploid secondary spermatocytes. The secondary spermatocytes will then undergo a second round of meiotic divisions to form the spermatids. The spermatids, again, are haploid, having only 23 chromosomes. And finally, the spermatids are going to mature into functional spermatozoa. Remember, the spermatozoa are the cells that have the tails, the ones that can actually do the swimming. Now, the spermatozoa aren't actually ready to fertilize an egg at this point. They're going to undergo a process of maturation within the seminiferous tubules and eventually be stored in the epididymis for up to several months before they're ejaculated. So through the process of spermatogenesis, we should get a total of 4 spermatozoa for every spermatogonium. Now, the spermatozoa has a couple different parts of anatomy that we need to know. The first of these is the head, and the head consists of 2 parts. The nucleus contains the DNA that will be the father's genetic contribution to the zygote. Also in the head of the sperm, we see the archosome. The archosome contains powerful hydrolytic enzymes which will help the sperm to penetrate the tough outer wall of the oocyte or egg. Down towards the tail, we can see something called the midpiece. And the midpiece consists of tightly packed mitochondria around a series of microtubules. And the purpose of the mitochondria, of course, is to extract energy from the seminal fluid and produce ATP. And this ATP will be used to power the tail or flagellum of the spermatozoa. Now, the interesting thing about spermatozoa is they live quite a long time inside the male reproductive tract, but typically only live 24 to 48 hours outside the male reproductive tract. So they have to be able to fertilize an egg fairly quickly, otherwise they're going to die. Now, another interesting factoid about sperm is that a typical male will manufacture somewhere around 400 million sperm each day. And this is quite an accomplishment when you think that females typically only ovulate 1, maybe 2 oocytes every month. Males are producing 400 million sperm every day. So next time you see your husband or your boyfriend and he's just laying on the couch like a bump on a log and doesn't seem to be doing anything, remember that he is working hard. He is producing 400 million sperm a day, and so you should probably let him rest. He's earned it. So now we're going to talk about the 5 hormones that regulate the male reproductive system. These include testosterone, inhibin, FSH and LH, and also GnRH, or gonadotropin-releasing hormone. Now let's start with the one you know, testosterone. Testosterone is a steroid hormone which is produced by the testes. It stimulates spermatogenesis, or production of sperm, and it also causes the development of male secondary sex characteristics. Now these include things such as body hair and muscle mass, and it's important to realize that the male secondary sex characteristics aren't present at birth. That is, we have a baby that's born and it doesn't necessarily look like a male. The masculinization process happens through the process of puberty, which starts around the age of 13, 14, somewhere in there. And at this point, a typical textbook will show a diagram like this. This is a very generic and lackluster diagram of the process of puberty. You can see somebody that is 8 years old and they don't have any hair, they don't really have any muscle mass, and by the time they get 15 you see that body hair is coming in, muscle mass is coming in, and by the time they're 18 they look like Chuck Norris. So in reality, puberty doesn't really happen like this in a linear fashion. Because it happens over a very long time period, we don't really tend to realize it's going on until we look at the photos of ourselves at different ages in our life. The wonderful effects of testosterone on the body. Let's go back and talk about the other 4 hormones that are important in maintenance of the male reproductive cycle. And these include the gonadotropins, LH and FSH. LH stands for luteinizing hormone and FSH stands for follicle stimulating hormone. But these were named principally for their functions in the female reproductive tract. They do have different and opposite functions within the male reproductive tract. So you probably remember from your endocrine chapter that LH and FSH were produced by the anterior pituitary, and that LH stimulates the testis to produce testosterone. FSH, on the other hand, acts indirectly to promote spermatogenesis. Specifically, it acts on the Sertoli cells, which were the mother cells, and it basically causes them to sequester androgens like testosterone within the cell to help to stimulate spermatogenesis and sperm maturation. And of course, we couldn't produce LH or FSH without GnRH. GnRH stands for gonadotropin-releasing hormone, and it's produced by the hypothalamus. GnRH stimulates the production of the gonadotropins FSH and LH from the anterior pituitary, and it does this through a negative feedback loop. Basically, high concentrations of testosterone, which were stimulated by LH, will turn off GnRH production and also FSH and LH production. So now that we've learned about the 5 different hormones that regulate the male reproductive tract, let's take a look at the hormonal axis that regulates production of testosterone from the testes. The first step will be production of GnRH, or gonadotropin-releasing hormone, from the hypothalamus. GnRH will travel to the anterior pituitary, where it will stimulate the production of FSH and LH. FSH will stimulate spermatogenesis as well as the production of androgen-binding protein within the Sertoli cells, whereas LH will stimulate the production of testosterone via the Leydig or interstitial cells. Together, ABP and testosterone will stimulate spermatogenesis within the Seminiferous Tubules. Once adequate numbers of spermatozoa have been formed, the seminiferous tubules will secrete something called inhibin. Inhibin has a negative feedback on both the anterior pituitary and also the hypothalamus. In addition, it's also important to point out that the testosterone is not limited to the testes. It's traveling throughout the bloodstream and eventually reaching its way back to the hypothalamus and anterior pituitary. Just like inhibin, testosterone has a negative feedback on the anterior pituitary and on the hypothalamus, and this helps to ensure that we don't produce too much sperm or too much testosterone in the body. And while you might think having too much sperm is not really a big problem for the body, having too much testosterone definitely is. If you've ever seen some of these bodybuilders who use anabolic steroids, you've seen the evidence of having too much androgen in the body. Not only are they very muscular, but they also can be very, very angry and very mean. And we often call this roid rage. And so excessive levels of testosterone in the body not only influence our behavior, but they can also put us at greater risk for things like heart disease. And so it's important that we have enough testosterone, but not too much. And that's why the production of testosterone is regulated by a negative feedback mechanism. So now that we've talked about hormone production and spermatogenesis in the testis, let's go up one step and talk about where the sperm are going to be stored. The sperm are stored in the epididymis. The epididymis contains a tightly coiled tube around 20 feet long, and this consists of a pseudostratified epithelium that helps to nurture the sperm and helps them to mature. This is also the area where we will store sperm. Sperm can be stored here for up to several months prior to ejaculation. Any sperm that are not ejaculated will eventually be broken down by the cells within the epididymis. Leading away from the epididymis, we have the first of our ducts, the ductus deferens or vas deferens. And remember that a duct is simply a hollow tube through which something's going to flow. In this case, this is going to be a hollow tube through which sperm are going to be transmitted from the epididymis into the vas deferens and eventually reaching the urethra. And this happens because the ductus deferens also contains smooth muscle cells which contract rhythmically to generate peristaltic contractions. And these peristaltic contractions will help to transmit the sperm up through the vas deferens and eventually into the urethra during the process of ejaculation. Now at this point we need to talk about a surgical procedure that you've probably heard of, and this is called the vasectomy. Remember, actomy means surgical cutting of, and so this is the surgical cutting or severing of the ductus deferens or vas deferens. And we do this as a birth control measure. On the left hand side you can see an intact testis with the vas deferens and also with the blood vessels and nerves leading away from it. So if we want to prevent fertilization, we simply have to cut and ligate the vas deferens, leaving the blood vessels and the nerves and so forth intact because we do want to have testosterone production. And so this is a very simple procedure and can be conducted on an outpatient basis, and if done properly, it's nearly 100% effective at preventing pregnancy. And again, because we're not messing with the testis and we're leaving the arteries, the veins, and also the nerves intact, this does not affect your virility or your sex drive. It only affects your ability to produce sperm. You might be asking, what happens to the sperm if they have no way out? Do they build up? Well, they tend to break down and the cellular components are reabsorbed by the Sertoli cells. Now, during the process of ejaculation, the ductus deferens will eventually transmit the sperm up towards the ejaculatory ducts. The ejaculatory ducts are formed by the union of the seminal vesicles and also the ampulla of the ductus deferens. An ampulla is just basically an enlarged portion, and this enlarged portion again has some smooth muscle in there to help rhythmically contract and force those sperm and fluid into the urethra. Now, the urethra is an interesting structure because in males it has both a reproductive and also a urinary function. And so there are three different parts of the urethra. The first of these is the prostatic urethra. This is the small bit of urethra that's located inside the prostate gland, which is an accessory structure used to generate seminal fluid. Next we have the membranous urethra, which is an even shorter portion which exits from the prostate through the abdominal musculature. And finally, the spongy urethra is the longest portion of the urethra that extends about 5 or 6 inches into the penis. So the urethra is going to be the site of ejaculation of semen, and it's also going to be the site where we urinate from. So the male reproductive tract has some unique adaptations to allow us to do both of these without interfering with one another. So now we're going to talk about some of the accessory sex glands of the male reproductive tract. The first of these are the seminal vesicles. The seminal vesicles secrete a yellow, alkaline, viscous fluid which contains fructose, prostaglandins, and clotting factors, and it makes up around 70% of the seminal volume. Now, the alkalinity is going to be important because it's going to help us to neutralize the pH of the female reproductive tract, which is quite acidic. Now, the viscousness or viscosity of the semen also helps to hold the sperm up in the female reproductive tract and simply keep them from dribbling back out again before we have a chance for fertilization. The fructose is in there is an energy source for our sperm, and the prostaglandins will stimulate smooth muscle contraction. And this is important because even though the penis gets the sperm fairly close to the egg, there's still several inches in between where the egg is and where the sperm are. And so the prostaglandins will stimulate something called reverse peristalsis within the uterus and fallopian tubes, helping to transmit those sperm closer to the site of the ovulated egg. Now, the prostate is also another accessory gland that helps to secrete seminal fluid. It makes about 20 to 25% of the seminal fluid, and this consists of a milky, slightly acidic fluid containing prostate-specific antigen. And we'll talk about that in just a couple slides. It also contains enzymes that will eventually break down the clot that was formed by the clotting factors manufactured by the seminal vesicles. Okay, our last set of glands are the smallest, and these are called the bulbal urethral glands. The bulbal urethral glands secrete an alkaline solution that helps to cleanse the urethra prior to ejaculation. So unlike the prostate and the seminal vesicles, these really don't contribute much to the seminal fluid. The fluid secreted by the bulbal urethral glands is actually secreted prior to ejaculation. That is, after the male has become stimulated, but before he has an orgasm and ejaculates. And this little bit of alkaline fluid helps to neutralize any of the residual urine in the urethra. And this is important because urine has a pH around 6, and so it's fairly acidic, and this could be very damaging to our very delicate spermatozoa. And so the bulbal urethral gland secretions neutralizes this acidity to ensure that our sperm are going to be in tip-top shape by the time they are ejaculated through the urethra into the female reproductive tract. So while we're on the topic of the male accessory glands, we should talk about some of the disorders associated with them, specifically the prostate. Now remember, the prostate was a gland that surrounds the urethra just as it exits the bladder. And this can be a particular problem because any type of enlargement can cause it to grow larger and basically pinch off the urine flow. And so it can make it very, very difficult to urinate, which is often one of the symptoms of all three of these disorders. The first of these is prostatitis. Prostatitis is inflammation of the prostate caused by a bacterial infection. Now, normally, just like any other bacterial infection, we can treat with antibiotics. But for some reason, the prostate seems to be fairly impervious to some types of antibiotics, and so we have to treat for a longer time than we normally would if it was another tissue in the body. Another type of disorder is something called BPH, or benign prostate hyperplasia. Remember that hyperplasia means greater than normal growth, and benign means non-cancerous. So benign prostate hyperplasia is an enlargement of the prostate, which is usually caused by androgens, specifically testosterone. And again, it can pinch off the urine flow, making it very, very difficult or uncomfortable to urinate. And the third disorder here is prostate cancer. This is the second most common cause of cancer death in males. And just like benign prostate hyperplasia, one of the symptoms of prostate cancer is difficulty in urination. So even though difficult urination is a symptom of both benign prostate hyperplasia and prostate cancer, it does not lead to a definitive diagnosis. In order to figure out whether you have an enlarged prostate or actually a cancerous prostate, the doctor will need to do a couple different tests. The first of these is simply to insert a lubricated finger into the rectum and use this to digitally palpate the backside of the prostate gland. And this can help the doctor to determine whether or not your prostate is enlarged and also whether it is irregularly shaped, which may be an indicator of prostate cancer. Now, if your doctor suspects that you might have prostate cancer, he may also do a PSA test. And PSA stands for prostate specific antigen. And this is an antigen that is normally produced by the prostate. It's part of the seminal fluid, but it tends to increase if the prostate becomes cancerous. And so we can use your PSA number as an indicator for how likely it is that you may have prostate cancer. Again, it's not definitive, and there's a lot of research now showing that high prostate specific antigen numbers are not necessarily cancerous and that we don't pay too much attention to them. The gold standard for diagnosing prostate cancer, of course, is going to be a biopsy of the prostate, which is not very fun. Now let's go back to BPH, or benign prostate hyperplasia. This was a non-cancerous enlargement of the prostate gland, which caused difficulty in urination. And so there's a couple ways we can treat this. We can remove the prostate, which definitely can be done, but more typically somebody's going to opt for a treatment that leaves the prostate intact and also leaves the nerves associated with the prostate intact, because these are the nerves that basically control the penis and also allow us to control our urinary sphincters. And so here you can see somebody putting a laparoscope through the urethra, and they're also inserting a small tool to burn or freeze away the excess tissue within the prostate gland and therefore clear up more of a cavity for the urine to flow through the prostatic urethra. We can even do something like balloon angioplasty, where we insert a catheter with a balloon at the end, and we inflate that balloon at the narrow part or stricture, and we basically enlarge that opening to allow urine to pass through more easily. Now, if your doctor finds out that you have prostate cancer, the treatment for that cancer is going to depend on how aggressive the cancer is. In some cases, prostate cancer can be very slow growing. So if you're a man in your 70s or 80s, your doctor might just take a wait-and-see approach. On the other hand, if you have an aggressive form of prostate cancer, we need to treat that right away because, again, prostate cancer is the second leading cause of cancer death in men. And so one of these treatments is something called a radical prostatectomy, that is, surgical removal of the prostate gland. And I know the slide here shows this jaunty little arrow, prostate's there, ooh, and then it's not. But it's not that simple of a procedure. For one, in order to get to the prostate, we have to make a surgical incision in the perineum. The perineum is the sensitive area of skin between the scrotum and the anus. And so here you can see in there, we've cut into the perineum, we've got these awful looking retractors that are pulling back the perineal skin, and now we can see some of the adipose tissue below that. And to get to the prostate, we have to go quite a ways in order to come in contact with it. And the radical prostatectomy itself is not a straightforward procedure. We go in there, we basically chip away chunks of the prostate gland, being sure to remove as much of the cancer as possible. And if you know anything about cancer, you know that we typically remove more than we need to. We typically have to go outside the area where the tumor is to make sure that we get it all, because if we leave any cancer behind, it can simply reoccur. And so a radical prostatectomy is a very radical procedure. Oftentimes it will destroy the nerves that control the penis and also control our urinary sphincters. As a result, people having a prostatectomy will oftentimes be impotent, and they will also have problems with urination. Basically, they will become incontinent. So fortunately, we also have some minimally invasive treatments for prostate cancer as well. And the first of these is something called LDR brachytherapy. And LDR stands for low dose radiation. And in this circumstance, we're going to take some small radioactive seeds and insert them into the cancerous prostate gland. And I know when I first learned about this procedure, I was like, if I ever get prostate cancer, this is what I'm going for. None of this cutting open the perineum business. I'll take these little bitty seeds inserted into my cancerous prostate. Well, it's not as simple as that. In order to insert these seeds into the prostate gland, we have to once again go through, that's right, the perineum. The picture down at the bottom of the screen, you can see what looks like somebody's buttocks, and then you can see this battleship-like grid that the guy has lined up on the guy's perineum. And so simply he's inserting these catheters through the perineum into strategic sites within the prostate gland, and through the catheters he's inserting those radioactive seeds. So it's still not a very pleasant operation. And finally, a third type of treatment for prostate cancer is something called cryotherapy. Remember that cryo means frozen or cold, and so cryotherapy is literally used to freeze kill the cancerous cells within the prostate gland. Now, just like with the LDR brachytherapy, this is going to involve inserting numerous pins through the perineum into the prostate. The other thing that you might notice in the picture is that large curling iron looking thing that's inserted up the rectum. That's actually an ultrasound probe, and it's used basically to gauge the placement of the cryotherapy needles in the prostate gland. Now, once they're in the proper place, the cryotherapy needles will be injected with a mixture of helium and argon to freeze certain sections of the prostate and kill off any of the cancer cells that are there. So while we're on the topic of male reproductive disorders, we should mention cryptorchidism. Cryptorchidism is basically the non-descent of the testes. Remember that the testes actually start up within the abdominal cavity very close to the kidneys, and this is the same location where the ovaries are located. And so what happens during normal human development is that around the age of about 4 to 6 weeks before birth, the testes will be pulled down into the scrotum by the gubernaculum muscle. If something interferes with this, we say that the testis is cryptorchid because it is maintained in the abdominal cavity. And if it's allowed to stay there, this will result in sterility for the male, and will also cause a greater incidence of testicular cancer. So assuming that your testes and also your seminiferous tubules and your seminal vesicles and prostate are working correctly, you're going to be producing a fair amount of semen in your lifetime. Now, semen is a nutritive transport medium for sperm. On average, each ejaculate is around 2 to 5 mils in volume, so not very large. When you think of pigs can produce up to 50 mils of semen in an ejaculation, that's quite impressive. But what you should realize is that the normal male is going to have somewhere between 20 million and 150 million sperm in each milliliter of that seminal fluid. Now, the seminal fluid is slightly alkaline. It has a pH of around 7.2 to 8.0. And again, this is to help neutralize the secretions of the female reproductive tract. The female reproductive tract is very acidic. It's a very inhospitable place for sperm to be. And so without this alkaline fluid, the sperm would be very quickly killed and deactivated by the low pH in the reproductive tract. So this alkalinity allows us to neutralize the acidity in the uterus and in the fallopian tubes and make sure that our sperm are going to be all that they can be. The other thing that's in the semen is antibiotics, which help to prevent infection of the sperm. And finally, there's also a lot of fructose in there, again, energy for the sperm, and prostaglandins. We said earlier that prostaglandins will stimulate smooth muscle contraction within the female tract. And this is important because the sperm, even though they can swim, they can't swim that far. And so the majority of the transit they do across the female reproductive tract will actually be through smooth muscle contraction within the uterus fallopian tubes that will help to propel the seminal fluid up towards the ovulated oocytes. And so that's the function of the prostaglandins. Okay, so now we get to the most conspicuous male reproductive organ, the penis. The penis is an organ of intromission. Now, if you don't know what that means, you might want to look it up. In this case, intromission is not the time between the first half and the second half of a movie. Intromission is actually the act of putting the erect penis into the vagina. And so it's a way to get the semen as close as possible to the ovulated oocyte. Now, depending on the length of the penis, this may only get it 3 to 6 inches closer to the ovulated oocyte. But this is very important because it drastically reduces the amount of sperm that human males have to produce. Now, let's think about animals that don't have a penis. Let's say fish. Fish, at least most fish, don't actually have a penis. And so as a result, they can't really get the sperm as close to the ovulated eggs as they would like. That means the chances of fertilization tend to be very, very low. So they basically have to use a different strategy to ensure that some of their sperm is eventually going to reach an egg and possibly fertilize it. And that strategy is basically to produce a lot of sperm. Here you can see a spawning aggregation of some fish, and that cloud you see, that's right, that's seminal fluid. And in this case, basically the male fish will go up to the female as she's ovulating her oocytes into the water column and basically just shoot a large mass of semen into the path of those ovulated oocytes. And that obviously is not going to be a very pleasant experience. I mean, particularly take a look at the fish in the background there. Don't know whether it's male or female, but it definitely looks a little bit nonplussed. And so you probably should just be grateful that male humans, as well as most other mammals, have a penis, because otherwise the act of sexual intercourse would be, well, a much more difficult and messy enterprise. Now that we know why we have a penis, let's talk a little bit about the anatomy of the penis. Now, the base of the penis is called the root, and the root consists of two structures. The paired crura, or basically legs of the penis, these help to anchor the penis to the pubic arch of the bony pelvis. And then in the center you can see the bulb of the penis. Covering the bulb of the penis is a sheet-like muscle called the bulbospongiosus muscle, which helps to secure it to the urogenital diaphragm and helps with ejaculation. The body of the penis consists of the skin and the underlying erectile tissue called the corpora cavernosa and corpus spongiosum. We have two corpora cavernosa and one corpus spongiosum, and basically these are the structures that engorge with blood and allow a man to get an erection when he's sexually stimulated. Now at the tip of the penis we have the glans penis. The glans penis has the opening for the urethra through which we're going to urinate and also ejaculate, and the glans penis is also important because it contains the highest number of sensory nerve endings in the male reproductive tract. And so the glans penis is important for receiving stimulation. And it is homologous or similar in development to the clitoris. And so whether you're a girl or a guy, you'll be able to recognize that the glans and the clitoris are both basically the centers that receive stimulation. Now, covering the glans penis, we may have a prepuce or foreskin. Now, in the United States, many times we will surgically remove the foreskin in a process called circumcision. Now, this used to be done just for a matter of aesthetics and for religious reasons, but now we're starting to realize that circumcision actually does offer some benefits. We found out that circumcision can greatly reduce your chances of becoming infected with the HIV virus as well as other sexually transmitted diseases. Now, we said before that the primary function of the penis was to serve as an organ of intromission, that is, to enter the female reproductive tract, and in order to do that, it has to be erect. And so an erection occurs when a male is stimulated, and this stimulation results in parasympathetic nerve impulses, which result in nitric oxide secretion. And nitric oxide is a very potent vasodilator, which causes the arterioles that feed the erectile tissue to dilate. And so we have a lot of blood that is reaching the corpora cavernosa, which you can see look like the eyes and that little shouty face. That's actually a cross-section through the penis, by the way. And so the corpora cavernosa will fill up with blood, and as they do this, it will actually pinch off the venous return from the penis, leading to a fully erect and engorged penis. Now, the corpora spongiosum isn't nearly as large as the cavernosa, but it does become slightly erect, and the reason that it's important is because it makes sure that the urethra is not squeezed off by the corpora cavernosa, and so it holds the urethra patent or open. And this is really important because if we have an erect penis but we don't have a patent urethra, there's nowhere we can ejaculate through. So we have to make sure that the urethra is patent in order for the sperm to get out of the urethra into the female reproductive tract. And so when the corpora cavernosa are fully engorged, we will have an erection. And again, the erection is there to basically get the penis into the vagina so that we can ejaculate and get the sperm as close to the ovulated oocyte as possible. Now just remember that an erection is caused by parasympathetic nerve impulses. And what this means is that erections tend not to occur when somebody is stressed or when they're worried or things like that. Anything that potentiates sympathetic nerve impulses is going to make it less likely that an erection is going to happen. On the other hand, ejaculation is actually a sympathetic reflex. That is, it originates around L2 in the spinal cord. So during ejaculation, sympathetic nerve impulses cause peristaltic contractions of the seminal vesicles, ejaculatory ducts, and also the urethra to propel semen very, very quickly through the urethra. At the same time, it also causes constriction of the sphincter in the bladder, and this prevents sperm from basically going backwards into the bladder, which wouldn't be very beneficial, and it also prevents the guy from inadvertently urinating while he's ejaculating, which would also be a pretty big party foul, I guess. And so you probably have noticed this if you're a guy, or maybe if you're a girl, that maybe your husband is not able to urinate right after ejaculating. And that's basically because the sphincter to the urinary bladder is sealed off during and immediately after the process of ejaculation. So in the previous two slides we showed what happened in the male penis during excitement and also during ejaculation. But you should realize there's actually 4 stages to the human sexual response cycle, and these were described by Masters and Johnson. The first of these is called excitement. Now during the excitement phase we basically have stimulation that could be visual, auditory, it could be tactile, whatever it is, it's causing parasympathetic nerve impulses which are basically causing vasodilation within the arterioles that are feeding the corpora cavernosa and spongiosum, and this leads to an erection. Now once the penis becomes fully engorged, we'll basically transition into the plateau phase. The plateau phase is when we have a fully erect penis, but we haven't yet ejaculated. And during this period, which can last for several minutes, the Cowper's glands, or bulbourethral glands, will begin to secrete a little bit of that fluid that will rinse out the urethra and neutralize the acidity of any residual urine. The other thing that will happen is you notice the testes have been pulled up close to the body, and so this prevents the testes from being injured during sexual intercourse. And finally, we're going to have the orgasm. That's at bottom left. Here we have the rhythmic contraction of the vas deferens, the seminal vesicles, and also the urethra, which will propel the sperm and seminal fluid out of the penis into the female's reproductive tract. And then finally, we're going to reach the period of resolution. This happens very, very quickly in males. Basically, after ejaculation, there's a period where the penis will begin to drain of its blood, the arterioles will begin to constrict, and the veins will basically dilate, allowing the blood to move out of the penis back into the body. The other thing that will happen is the testes will once again begin to descend away from the body, and during this period it's typical for males not to be able to achieve another erection for some time. There is a refractory period between which a male cannot have another erection or another orgasm. Now, this refractory period depends on the age of the person. If you have a very young male, he may be ready in another minute or two, whereas an older male may take several minutes to even hours to be able to achieve a second erection. Now, while we're on the topic of erections, we should talk about erectile dysfunction. Erectile dysfunction is a disorder that affects greater than 50% of men over 40 at some point in their life. And it has multiple causes. It can be caused by diabetes mellitus, arterial sclerosis. Certain drugs such as antidepressants can cause erectile dysfunction, as well as heavy alcohol consumption. The other thing is we can just have a generalized neuropathy that is interfering with the sensory information traveling from the penis to the brain and back again, and that can also lead to erectile dysfunction as well. And so previously the treatment we had for erectile dysfunction was a penis pump, where basically you would put this tube over the penis, you would inflate it, it would draw a vacuum in there, and basically that would eventually lead to an erect penis. Now, this can make foreplay and so forth a little bit awkward. A more recent pharmacological treatment is the development of PDE5 inhibitors. These are drugs which basically lead to the production of nitric oxide within the arterioles of the penis, and they allow them to dilate, allowing blood into the corpora cavernosa and corpora spongiosum, and they tend to be very effective at causing an erection in somebody that's been adequately stimulated. And so these treatments were initially developed to treat hypertension in men, and they didn't tend to work very well. But what we did find out in the research studies is that some of the men came back and said, well, my blood pressure hasn't gone down, my angina hasn't gone away, but things are starting to happen that, weren't happening before for a long time. And so drugs like Viagra and Cialis have had a very brisk sales on the market. Just last year, there were over $5 billion worth of erectile dysfunction drugs sold on the American market alone. And so it's a very popular and very lucrative drug. Now, interestingly enough, Viagra and some of the other ED drugs are also used to treat other types of conditions. Some people have used it to treat pulmonary hypertension, that is hypertension within the pulmonary loop, and other people have used it to treat the cerebral edema that occurs with altitude sickness. So people that are hiking up at very high elevations can take Viagra to prevent this potentially deadly brain disease that happens to some people at elevation. And finally, another use that I just read about the other day is that some florists will actually take a little bit of Viagra and put it in the water of the cut flowers they are trying to keep alive. As it turns out, they will keep those flowers erect for about another 6 days beyond which they would normally last, and so it's been a real boon apparently to the flower industry. Go figure. This concludes our lecture on the male reproductive system. If you're interested in learning more about the sexual response cycle, either male or female, you can click on the 2 links below. The one at left is a link to a short little YouTube video. It's kind of grainy about Masters and Johnson. Masters and Johnson was a team of researchers in the 60s that first described the sexual response cycle in both males and females. And they did this by basically watching and recording the various things that happen in the body during sexual arousal and eventually orgasm. And so they had all kinds of transistors and conductors hooked up to their patients. And some of the things that they measured in their patients during the different stages of sexual arousal was, one, the heart rate becomes elevated, the respiration rate becomes elevated, the eyes initially become very, very dilated. And so we learned all these things about arousal that we never knew before. And again, this was happening in the 1960s. This was after Kinsey, but before the public had really become receptive to any type of sexual research. So very, very fascinating work they did on the human sexual response. Now the link at the right side of the page will take you to a short video produced by Discovery Channel, and it's basically on the anatomy of arousal, and it shows what happens in the male and female body during sexual arousal and eventually orgasm. And it's only about a 6 or 7 minute video, but it tends to be very interesting and very easy to understand. Now at this point, I'd like to offer some extra credit to those of you in my online class.